بسم الله الرحمن الرحيم، والصلاة والسلام على أشرف المرسلين سيدنا محمد وعلى آله وصحبه وسلم. اللهم لا سهل إلا ما جعلته سهلا، وأنت تجعل الحزن إذا شئت سهلا. اللهم انفعنا بما علمتنا وعلمنا ما ينفعنا يا رب العالمين. أهلا بكم في مراجعة 11 أدفانس العام 24-25 العام الترم الثاني. لو جينا شفنا الهيكل، طبعا أنا عشان تبقوا فاهمين الموضوع مترتب إزاي. مترتب، أنا رتبته حسب صفحات الكتاب. يعني قبل كل صفحة أنا حاطط الهدف. ادي مثلا صفحة، أول صفحة اللي هي صفحة 74، حاطط الهدف اللي جاي منها. اوكي، وبعد الصفحة في بعض الأسئلة من السنوات السابقة. هذه أسئلة لها علاقة بهذا الجزء. نستعين بالله سبحانه وتعالى ونبتدي ونقول بسم الله الرحمن الرحيم. السؤال الثالث أو page number 3 in MOE. It's called about the figure 1 and 4 in the same question. It has the two pages 74 and 77, so that we going first to 74 and see what is the curriculum in this. 74 we will found the genetic engineering, what the meaning of genetic engineering, and the GFB day, okay, and what is used. This is the main point pages, okay. The main point is this page and this figure, okay. Be careful about it. If we're going to see some question from last tests, we found one like that. He asked about what? He gave me or give us this picture and tell us the image below represent an experiment where a gene from an ocean jellyfish that creates bioluminescence light has been inserted into a mosquito. The mosquito with the jellyfish gene gives off green light. Which of the following explain what scientists have accomplished? Okay, if we see here, we see many choices. As we know, this is genetic engineering. Okay, the gene of the mosquitoes were manipulated using genetics. In the definition of genetic engineering, we say we say 4 words. Its meaning genetic engineering happen. Number 1, manipulate. Number 2, modify. Number 3, change. Okay, all of this is about modifying a DNA or the fourth word is using, okay, to manipulate or using forward like that. Okay, so this is the correct answer. Let us see why the other answer is not. DNA fingerprints. It's not about fingerprint. A new form life was created. It's not about cloning a new life created. Natural selection It's not common or not attached with natural selection. After that, we're going to the next question. Okay, the next question is asked about which of the following organism has been genetically engineered to improve the food supply and human health. So that we will choose the one of the examples here. It's contact with improved food, number one, and supply health for the human. Goats, maybe. Chickens and trucks, maybe. The roundworm, maybe. But he tell us the last one, transgenic livestock. Transgenic livestock. We will choose this one because it has a genetic engineering change. Okay. The goats is normal goats. Chicken and the trucks, it's normal because he didn't tell us about anything about it. And this organism is used in lab of genetic engineering many times, so that when he changed this and asked about which organism is using more and more in lab, we will choose this one. Okay, let us go into the next. Here, let me make it bigger. Okay. Which of the following organism has been genetically engineered to used in research laboratories? This is what I say before 2 seconds. Around the world to study diseases and develop ways to treatment. Okay. Let us see which one. Goals? No. Chickens and turkeys? No. Fruit flies? Maybe and maybe not. Transgenic livestock? No. So that he changed this one, the roundworm, by this one. Fruit flies, so that we will choose this one. Okay. After that, we're going to the next question, which is an example for transgenic organism. When he asked us about this question, transgenic organism, we will see or look for which one has an award about genetic engineering. So, German Shepherd to identify explosive? No. Hybrid race? No, this hybridization, maybe this is natural selection. Okay. Bring me mints, radish to kill garden pests. Okay. Nothing else because it's bring. Okay. Chicken genetically engineered, okay, engineered to resist a disease. Okay, this is the true choice. Let us see the next question. Which of the following organism has been genetically engineered to improve. Again, this is another question, which is again, okay, transgenic livestock. Okay, let us see the page after that. Page after that will be page number 75, and in the MOT, okay, sorry, EMO, he asked about the figure 2. Figure 2. Figure 2. And this is the page from the book. And this is picture 2. And I want to give you an advance. When he asked or put a figure, you must read what about this in the curriculum, so that this figure number 2, okay, where this is mentioned here, maybe in this page here, I think. Okay, no problem. As we see here in the picture, it's called or study about the restriction enzyme. We have two kinds of restriction enzyme, one called as sticky end restriction enzyme, and another one called as a blunt end. If we say if we see two fragment of DNA, and it's not completed like that, okay, it's cutted by restriction enzyme sticky end. But if we see a fragment like that, it's completed, it's meaning it's cutted by blunt end, not by sticky end. Okay, right. The famous one of sticky restriction, restriction enzyme sticky end is called as EcoRI or EcoR1, sometimes called like that, sometimes called like that. Okay, let's see some question about it. Here, the first question: which of the following enzyme cuts the DNA and created fragments as shown in below? This is sticky end, so that if we want to see the child, RNA polymerase, no. DNA ligase, no. Okay, reverse transcriptase, no else. EcoRI enzyme or EcoR1, okay, is choose or the correct choose. Okay, another question or another page. Yes, in this page we have two questions. Question number 2, question number 5. This is the number of question, and this is the objectives, and this is the number of aim or objective. Okay, and this is what he wants, all the picture, all the page, or he asked about the figure. We here found two questions on the figure number 3. Let us see which is the figure number 3. This is the figure number 3. Let's study the gel electrophoresis. And gel electrophoresis, you must know, number 1, where you put the sample. This is the first. Okay, the technique for separated the fragments of the DNA which cut it by the instruction enzyme. Okay, and what is the base for this technique? It's arrange the fragment as its size from biggest to smallest. Okay, the biggest is near from minus and the smallest is near from plus in the cell of gel electrophoresis. So that when we see like this lines and this tool, we immediately say this is the gel electrophoresis. Let us see some question about it. This one of the question here, as I say before, the big fragment will be here, and this is arranged by the size until we going to the plus, the biggest, the less big, less big, less big, until we found the last one, which is the smallest. So that the smallest here, the biggest here. The question is asked about the figure below shows the DNA fragments pattern after the paste gel was placed in the electrophoresis taken tank, and the current was turned on which of the following letters represent the band that has smallest DNA fragment. Okay. So that the true choice is D, because D is the smallest and the biggest is A. Okay. Let us see another question and clearly question than this one. I'm sorry because I didn't see it well. Okay, I have some problem in my vision. I'm sorry. Right. When the loaded gel is placed in the electrophoresis tank and the electric current are turned on, the DNA fragment is separated. What processes does the figure below shows? As we said before, the gel electrophoresis, so that we will see gel electrophoresis is the true choice. Another question, the third question is what the process does is the same, gel electrophoresis. Okay. Let's see else another page and another. This is question number 3. Question number 3, we mentioned it before in the first page. It's contact with 74 and 77 page. Let us see the first. It's the same, so that I put it here again. Okay, the first slide here, you will see the same, okay, figure and same pages, so that I put page number 47 and this is the page number 77. Where is 77? This is a 77. And 77, we see the recombinant DNA and the plasmid. And please be careful about this figure, because it's coming more and more and more and more in many tests, and you must remind every word in this page. Okay, what is this called? What's this called? What it mentioned here? What is mentioned here? And what is mentioned here? Okay, and what's the name of the enzyme which stick or join the DNA from organism with the plasmid is called as ligase. Okay, don't forget any word here. Okay, let us see some question about this one. As I say before, if you see here, this is picture. Recombinant DNA is created by joined DNA from two different source, which letters of the following refers to genomic DNA, and the illustration shown below. If we return to this one, it will be genomic DNA. Okay, so that this is will be genomic DNA. The true choice is B. True choice is B. Okay, first question. We're going to second question. Genomic DNA, else it's B. Okay, third question. Here he asked about which enzyme is used to what, and the region indicated by the letter C. He asked about the letter C, which joined this gene from genomic DNA with the plasmid which coming from bacteria. Okay, what the enzyme join them here, what's the name? As I say before, it's ligase. Okay, when you forget it, think about your leg, ligase. Okay, your leg. Tamam. Okay, it's DNA ligase joining enzyme. Let us go to another question. Question number 4. Question number 6 is the same page. 78 as all. Okay, and 78 figure 5. Which figure 5? This is the figure 5. As I say, and recombinant DNA, be careful about this picture, else, and every word must know about it and know what is the meaning of AMB. AMB is a gene for a resistance for ampicillin. Why they put it? Because when they wanted to put this recombinant DNA inside the bacteria, some bacteria is taken and some bacteria didn't taken. Okay, and when they put the bacteria in the media which has ampicillin antibiotic, the bacteria which didn't take this resistance or the plasmid, okay, will die, and the other one will stay alive. This is the purpose from put this A and B. Okay, didn't forget every word here, please. They asked you about anything of this one. Okay, don't forget this page have two questions, one in the figure and one in the curriculum, other curriculum in this page. Okay, let us see an example of last question. As I say, okay, here 1, here 2. If we see what's 1 and 2, okay, this called as transformation because we has a transformed bacteria, and this is called, I think this is 2, yeah, this called as what? A replication. Bacteria also copies recombinant plasmid DNA. Let us see what he asked about. Which of the following do number 1 and number 2 refer to? He asked about number 1 and number 2. What happened? This transformation, and this is cloning, or as we will see now, transformation and cloning. Okay. Another question about this figure. What is the process that scientists use to produce large number of recombinant DNA molecules as shown in below? That's cloning. DNA cloning. Okay. After that, we're going to another page and another question. Question number 8, figure number 6, and page number 79. This is the page number 79. As we see, this page number 79. Let's study the what? Study the sequencing of DNA, sequence. Didn't forget we put in sequence the material we put in the sequencing. It's number 1, a fragment of DNA, okay, which want, or the DNA which want to know the frequency of DNA of nucleotide or nitrogen base on it. This is the first. Number 2, we will put a nucleotide or base, nitrogen base, with normal, and nitrogen base with an fluorescent day, like we see here. The G is blue, C is green, A is red, and T is orange. And we will put DNA polymerase and primers. This is a primer, and this is the DNA. And this is the DNA. Yes, we want to know the sequence of it. Okay. Right. Let us see some question about it. If we want to read the what is written here. I can't read this one. My vision is not clear for this one, but I know this one is called as gel electrophoresis for reaction mixture include an unknown DNA fragment and DNA polymerase, primers and DNA polymerase, the 4 nucleotides and different tagged nucleotides. This is called as tagged nucleotides. Okay. But I didn't have any question from last text about this, so that you must read this one well and see this picture and see and know everything about this picture, so that we are going to another question number 7. Let's contact with 82 page. This is the 82 page. Okay, it's asked about or study about what? About transgenic bacteria. In transgenic bacteria we have more things which is a benefit for human, so that you must know about it. Okay, let us see some question, example of question. transgenic bacteria can be used to do all the following. Expect one. Okay. Which one? Let's see. The non-coding regions of DNA are identical, each individual. The non-coding region of DNA can create proteins. The coding regions of DNA cannot create proteins. This all is true, but one only is not true. The non-coding region of DNA are unique to each individual. This is the choice. You must ask it again. Transgenic bacteria can be used to do all the following. Okay, expect one. Okay, this is the question or the truth. Okay, next question. Which is example of transgenic organism? I think this is a repeated question. Okay, transgenic organism. We see it before. Let us see another page and another question. This question 9 about the 84 and 86. This is 84. Okay, this is 84. It's about sequencing the genome. Okay, and you must study all this picture. There are some questions about this picture. Which of the following statements about the human genome is false? One of this one is not true. Let us see which one. The human genome contains long stretches of DNA with no codon function. Okay, it's true, but he wants the false. Okay, the human genome contains approximately 3 billion base pair. That's true. Else, the human genome was sequenced by scientists from around the world. It's true. Else, but he wants the false. Okay, the human genome contains nucleotide sequences that all code for proteins. This is wrong, and this is he wants the wrong or the false. Okay. After, sorry. Another question: which process is applied to identify mutations of errors in the DNA molecules? This is called as DNA sequencing, not recombinant DNA, not genetic engineering, no polymerase chain reaction. All this one is not true. This is the truth. Another question on another page. Sorry. Here, number 14 is asked about 85. 85, this is the page 85. It has a comparing genome, identifying genes, and bioinformatic. But I didn't have a question from last tests about that, so we're going to the next one. Number 8 and number 10. 9, sorry, and 10. The 9 and 10 is asked about the 44 and 84 and 86. 84 and 86. Okay, so you must study 84 and 86 well. This is 86. And 86 is DNA typing, or as called fingerprint, is the same name for these two. You must know what's its meaning, DNA typing. Yeah, return again. Okay, we return. Okay, if you see here, this is the used or that, okay, victim to identify the victim and the criminals, and also to exonerate or free people who had been wrongfully. Okay, as I say, you must read it well and see what is the benefit for this user. Let us see some question. Which statement about the DNA fingerprint false? We see here there are 4, and this is a true. Identify single nucleotide polymorphism, the SMP. Okay. Why this not true? Almost unique to each individuals. Okay. He asked about what is false. This meaning the other 3 is true. Almost unique to each individual. Yes, fingerprint or DNA typing, it's almost unique to each individuals. Identify individuals who have committed crime. Yes, it's using for that. It analyzes the longest reach of non-coding regions of DNA. Yes, so that this is 3 is true, but he wants a false. This is a false. Okay. Right. Let us see another page and another question. Sorry, this page. Yeah, it's number 13. Question number 13, and the page number 88. It's all, so that you must study this picture. Sorry, this page all. It's about DNA microarray. Okay, didn't forget it. And after that, page number page number 89. This is 89, and figure number 13. The figure number 13. It's complete the idea of microarray, and you must know the technique of microarray and how it's happen and the steps of microarray from they taken the DNA, and after that using the restriction enzyme to cut it into fragments, and after that they sorry, they using the I extracted the mRNA from it, and after that they using the reverse enzyme to make a DNA from RNA, which called is cryptase or DNA cryptase. Okay, and after that they mix the two kind of DNA, but the DNA new which coming by this reversible process is called as cDNA. One of them is tagged by the red color, and the other one is tagged by the green color. And this process or this technique is used to differentiate between the DNA in normal cell and another cell. Okay. After that, they put it in the microarray slide and see what happened after that. Okay, let us see another question, because I didn't have last question about this one from last years. Okay, figure number 11, question number 11 and figure 15 and picture number 90. Picture number 90. There are something, I'm sorry, I remember it now. Sometimes he bring this one and asked you about the ratio of red spots, so that you will count all the spot, and after that calling the red one and divide the red one by the all. Okay, you will get the ratio of the red. You can use it if he asked you about the green, else, and yellow, and so on. For 90 pages, figure number 14. Figure number 14, you will see the application of the human genome, this one. Okay, this one is for SMB, and there are one equation on it. Okay, and didn't forget the HapMap project. All right, if we see here this picture, and he asked the graph below an illustration of the HapMap project, which of the following represents the primary goals of the project? The primary goal of the project: a map that sequence of nucleotides and human DNA? No. Create a database for genetic biological information? No. Catalog the genetic sequences of individual DNA? No. Else. Identify genes that causes serious human disease? Yes. This is a primary goals of the project HapMap. After that, we will go into the next page and the next question. Question number 15 and page number 99 and 100. This is the 99, we now we will enter the second unit, the Darwin's voyage. Okay, Galapagos Island. You must read this picture well. This pages well. Number 99 and the number 100. Okay, in 99 we have a last question. And this question, he put a map like that and asked it, the figure below shows Darwin's journey and the animals has studied as at Galapagos Island of South America, which was the first conclusion Darwin made soon after return from Galapagos Island. If we see here, varied among Galapagos birds result from selective breeding? No. New species of finches could emerge through small ancestral change? No. Wild finches diversity explained the breeding of domesticated animals? No. Natural selection, because when he returned, he thought the main things in his theory is based on the natural selection, the evolutionary mechanism for species change. Okay. Another question in this part: which of the following terms best represent the cumulative changes in groups of organisms through time? This is the definition of evolution. Okay, it's the evolution. Okay. Next, this is number 100. 100, we have two questions. The last one here, this is the last one, number 15 and number 17. So in page 100, we have two questions. Okay, as you see here, this is 17 and number 10 for all the page. In page number 100, we will see what? From knowledge, natural selection, and Darwin's continued his studies. Okay, natural selection, just the origin of species. Okay, here, according to Darwin, what is the result of natural selection in a population? No change? No. All the individuals acquire the same characteristics? No. The environment adapts its to meet the needs and traits of the population? No. The population evolve with favorable traits becoming more common in future generation. Okay, this is the true and the other is false. I give you an advice because we study the natural selection of and the theory of Darwin. You must study this page here about the natural selection steps. Variation, this is the first. Heritability, this is 2. And overproduction, this is the third. And the last one, reproductive advantages. Okay. Let us see one question about that. Sorry. Darwin's preferred to process of promoting certain traits by breeding members with those traits as artificial selection. Artificial selection, this is the true. Okay, next. Another question with another page. Question 16, question 18, and page 102, 102. This is the page number 102. Okay, the question here, one of all the page, and the other one is table 1. So be careful, there are 2 questions in this page, one from all the page and one from this table. This table is study what? Let us see. Basic principles of natural selection. Individuals in population show variation. Okay, as I say this picture, okay, it's mentioned here in this table. Number 1, the variation. Number 2, inherited. Number 3, more and more overproduction. It's meaning overproduction. More young can survive. Okay, number 3, the reproductive. But study this this table well. Okay, and the other knowledge in this page. Okay, let's see the next. Here, number 19, question, page number 104. This is 104. It's called about Archreptus, okay, and comparative anatomy. You must study this page well. Let us see some question here. And question, this is art's rendering of Archaeopteryx shows that it shares many features common with. Number 1, the bird and the mammoth. Mammoth is the ancestor of the elephant. It's no. The amphibians and reptiles, between amphibians like toad or like amphibians like what like frogs, okay, and reptiles like snake, okay, the shrunk, the reptiles and the mammals, no else, the birds and reptiles, yes, the birds and reptiles, because it's as immediately, sorry, middle fossil between the ancient and the original. Okay, another question, I think it's the same. The birds and the, yes. Okay, the last question in this. Scientists study evolutionary relationships have found hundreds of thousands of transitional fossils that contains features shared by the different species. Which of the following are newly evolved features that don't appear in the fossil of the common ancestor? Okay. We have three original features and ancestral features and transitional fossil or transitional, yes, transitional fossil. Okay, the transitional fossil have some trait which is in the ancestral fossil, and the transitional fossil have else another trait which is in original organism. Okay, so that it's called as derived traits. This traits which is in transitional fossil and original organism is called as derived trait. Let us see another one. 21, table 2 in page number 105. This is 105, and this is a table. It's called about vestigial structure. We have 1, 2, 3 through 3 vestigial structure. Wisdom teeth in human, and wings in emo or kiwi, okay, and tailbones in humans, or yes, in humans, 2 in humans or 1, okay, so that you must study this table well, and if there are something mentioned about here like that, okay, from here you must study all this else. Let us see a question about it. This question asked about these 2 pelvis, okay, and I think in the snake, the image below, pelvis super are reduced forms of the leg bones of these snakes. Yes, ancient ancestors. Which term best to describe pelvis spores? Okay, its meaning that's vestigial structure. Vestigial structure. Okay. Let us see else another question. 20. That's figure 10 and 108. This is a 108, and this is a figure 10. Animals is called as a mara and English rabbit. Okay. Figure 10, the mara exists in niche, similar to that the English rabbits. What's say a kind of adaptation? Where is it mentioned? Here, no. mention for it. Here, no mention of it. As I say before, you must study the picture or a figure and see which it mentioned and study the knowledge about it. Okay? Let us see if there are a question about it. No, I didn't have any question for it. So that we will go into the next question, 22, page 109. You must study all the page. It's called about the camouflage and mimicry and mimicry. Okay, we will see some question here. If you see this picture, you will see a rabbit. Okay, as this is rabbit in ice, so that is called as camouflage. Yes, camouflage. And another here picture, you see the sticky animals or sticky rabbits, study the photo of the walking stick insects, sticky insects below, and decide what kind of morphological adaptation is shown. It's else as a camouflage. About mimicry, here is two kind of snake, and the figure below, the photo of California king snake shows which king of morphological adaptation. It's mimicry. I added this page because to know what mentioned in the next page, you must know or read this page. Okay, and sometimes they bring a question like that. Even though the number of owls, would the ratio of gray to the red owls remain the same? Which concept of the following apply to the figure below? This is the Hardy-Weinberg. They put, yes, it's called as Hardy-Weinberg principle. Okay. '24 and page number 112. This 112. It's about the Hardy-Weinberg. You must study this well. This table. If this 5 station or 5 case happen, the Hardy-Weinberg principle will happen, and the population will be balancing or sorry, yeah, it will be balancing. And if anyone from this case not happen, okay, this meaning there are an evolution will happen in this population. Hardy-Weinberg, he has two formula. Okay, let us see an equation, but before this equation we're going to whiteboard to know what is the Hardy-Weinberg principle. Hardy-Weinberg he say he has two formula. Formula number one is called as B plus Q equal 1. And the second one is called formula is B power 2 plus 2B Q plus Q power 2 equal 1. Okay, what's the meaning of B? B here, its meaning B equal the frequencies of dominant trait. Dominant trait, okay, frequencies of dominant trait. An example, okay, if we have an D capital, we will count D capital, and after that we will divide it for the full number. Full number of the alleles. Okay, this is the B, and the Q is the same as frequencies of recessive excessive trait. This meaning the small divided to the full number, so that we know now what is the B and what is the Q. So that I write here dominant, I write here recessive. Okay, طبعا it's frequencies of dominant and the frequencies of recessive. F, where F, yes. But what the meaning of this one? And what the meaning else of this one? And what the meaning else of this one? This one is meaning the frequencies of D S multiply and frequencies of D. Okay, and here this meaning the frequencies of recessive multiply the frequencies of recessive. Okay, and what this meaning? This meaning the two multiply in frequencies of D multiply in frequencies of recessive. Sorry, I want the color is the same. Sorry. Okay, here frequencies of D frequencies of D, and here frequencies of recessive. But be careful. This equation is for alleles. And this equation for genes. Okay? And if he asked us about any one of this one, if he asked about only allele, B, this meaning you will use the first. Okay? If he asked about allele, or طبعا هنا, or Q. Okay? If he asked about like that, G capital, G capital, this meaning he wants the B square. And G small, this meaning he wants the Q square. يبقى for homozygous. Okay? Homozygous dominant and homozygous recessive, this meaning Q, and heterozygous, G capital, G small, it's meaning he wants the two be Q. Now everything is very clear. If he tell me about the P, he wanted the frequencies of the dominant. If he wants the Q, it's meaning the frequencies of recessive. Okay. This is dominant, sorry, this is the recessive, sorry. I must read here R. Recessive. Okay. If he asked about G capital, it's meaning the second formula, and he wanted the P square, it's meaning homozygous. I write here homozygous. Okay. If he write that, it's meaning G small, it's meaning homo recessive. Okay. If he want heterozygous, meaning G capital, G small. Sometimes he asked by the name, not by the letter. Be careful about that. He tell you bring the ratio of homozygous, or the ratio of heterozygous, or the ratio of like that. Okay? Let us see an equation and solve it with the whiteboard. Okay? Here, an equation. He tell us based on, let us make bigger, okay? Based on the table below, use the Hardy-Weinberg equation, and he gave me the equation, excellent, to determine the equilibrium frequency of the dominant whole T capital. This meaning he wants B plus, B square. Genotype in the population of bee plant. This is a bee plant. He tell me bee plant population is 100. Okay, this is the all. So I write it's 100 as old. Okay, But now I want to make the frequencies of T capital, and the T capital T small, and the T small T small. Okay, he want the frequency to be square. This meaning I make like that. Maybe one of us call its T capital is 40, so that multiply it on 100, it will give us 0.4. But if we see 0.4 is not true, 0.4 not true, it's 0.36. How this happened? Let us see how this happened. So that I'll take this one of this picture, okay, to the to the whiteboard to solve it with, as we see now. Okay, now I take it and I'll put it here, make this one here, okay, like that, and no problem here, and make it bigger, okay, and let us to start. Let me bring my calculator. Number one, let us see the to count the fragments of the alleles. Okay, number one, here we have for T capital, it's 40 individuals. This meaning every T, we have 2 T equal 40. This meaning every T it has 40. To count 1 T, it's meaning we will multiply in 2, because we have 2T, this meaning 80. This is the first, okay? Right. Second, and let us make, okay, no problem. Second one, T capital, T small is equal how much? It's 40 else. This meaning T capital is 40 and T small is 40. So that I'll plus here a 40 for T capital. But this T capital only. Okay? And make like that. Going to T small. T small. We here found T small is 20. T small 20. This meaning every T is 20. So that it will be 20. Let me, 20 multiply 2 equal 40. Okay, 40 plus this 40 else, and we will make like that. Okay, let us see. This meaning a T capital equal 80 plus 40, so meaning 120. 40 plus 40 equal 80. 80 plus to make the total number of the alleles, okay? We will count now this 2 to bring the frequencies of P and Q. 0 plus 0 is 0. 2 plus 0 is 10. 0 and put here is meaning 200. Now to count the B equal this one 120, okay, divided to 2000, and the Q, it's for the recessive, it's equal 80 divided to 2000. This equal, let's using the calculate, multiply 200 equal 0.6. Okay, let us see the else one. 80 divided 200 equal 0.4. Okay, now he want what? He want the B, sorry, let us see. He want the T capital, not this T capital, he wants this one. This meaning the frequencies of T multiply the frequencies T, so that we will write what? We will write like that, okay? T capital equal B square. This meaning the frequencies of T multiply the frequencies of T. It's meaning 0.6 multiply 0.6 equal, we using our 0.6 multiply 0.6 equal 0.36. If we return to the question, let us go into the question again. The true one is 0.36. okay. يبقى the tricks، خدوا بالكم يا شباب لو سمحتوا، the tricks انك انت لازم تحسب وتقوم بعمل الحسبة دي. اوكي، مش هو يضحك عليك يقول لك هات لي تي كابيتال، تروح انت رايح حاطط قاسم ل 40 على ما اعرفش ال 100، انا مش دعوه بالعدد ده. This number I didn't mention of it. Okay, you must be in Hardenberg, you must before everything count the P and the Q. If you want T capital, it's meaning B square. If you want T small, not this number, you must count the Q and after that make Q square. If you want the T capital, T small, this meaning you will write 2 multiply P multiply Q, and after that you using these numbers to bring the total number. I think it's clear now. As we again, I'll put the formulas in front of you. Okay, this is the formulas. I write it before. It's clear now. Okay, let us see the question. Also another question. This is the same question, I think. Okay, as we say, 3, 6, yes. And here another question. Which of the following is not condition of the Hardy-Weinberg principle? Hardy-Weinberg principle, they put 5 cases to the condition of them theory happen. The 5 conditions here, it's mentioned here. Yes, number 1, population is very large, must be very large. The population didn't have immigration or immigration. The mating must be random. The mutation does not happen or does not occur. The natural selection does not occur or does not happen. Anything else or reverse of this one, this meaning the evolution will happen. And this, so that if we see here the random mating does not occur, this is not a condition of Hardy-Weinberg. Another Hardy-Weinberg here, in a population of frogs, the allele green is G capital, has an allele frequency of, he gave me already the frequency, excellent, of B is 0.3, while the allele for brown skin color G recessive, G small recessive, has allele frequency is 0.7. Scientist wants to estimate the expected genotype frequencies in this frog population to understand how common each skin color genotype is. Okay, what is the expected frequencies of gene-skinned homozygous frogs? You want this one. This meaning you will take the frequencies, this frequencies for G capital, for B, and multiply it in itself. If you made it in your calculate, that's 0.3 multiply 0.3 equal 0.09. This is the true 0.09. Okay. Let us see else another question. Here I think this is the same, but the number is change, and he asked it again for T capital. Okay, you can try it by the same way, and it will be the same number. Okay. Here this question about what he asked about. Asked what is the percentage of the, I didn't see well, sorry. What's the percentage of the melanic mouth population in 1994 in Michigan. Okay, let's see. Recent هذه Michigan is the green one. He asked about 1999. This 1000. Where's 1099? 199. sorry 90 كم 100 sorry 1994 here. Okay, if we're going and see the number, it was about what 90, I think it's 90, 80, sorry, how does this happen? Let us read. Here, sorry, I'm sorry, that's about 18, yes, 18, okay. Question number 25, the page 114 and figure 16. This is the page, okay, and this is the figure. You have three kind of natural selection: supplies natural selection, with distributional natural selection, with destructive natural selection. You must know what is the difference between the three. Okay, and read this page well. This is a question on it. If you see this picture is destructive, use the figure below to answer the question. Which kind of the selection does the graph best? Okay, it's destructive, as we addressed. Destructive selection, because it has two tunnel. Okay, else this question else is destructive, and here, sorry, there are an question here. Let us see, not this one, yes. Here, how does destructive selection influence genetic diversity within a population? So that you, as I say, you must read every one what's its meaning, not the name only of the figure. It favors individuals with extreme traits at both ends of the rank. Okay. We see this and this question. Let's see. Like, hypothesized environmental conditions could lead to shift in the variation of the snail population. Which type of the natural selection? He tell me shift, this meaning directional. Okay, so that it will be directional selection. Which of the following is not a principle? Darwin's theory of evolution by natural selection. Similar trait of the species. This is not a theory of the Darwin's type. Sometimes they bring a question like that. Be careful about this question, because I'm sorry, I think we transferring to another question. Yeah. Here, which of the statement about the turtles below will be part of an example for the turtles' evolution based on natural selection? This one, turtles with don't shells have more young than, let me make it bigger, you can read it, than turtles with the flat shells. Okay. another page. It's 23 question. And this is the last page, I think. Figure 18, 160. Okay, this page and this snakes, which it's study about the kind of natural selection is called as disruptive selection. Okay, this is an example of disruptive selection. In the end, we have some questions, an extra question. If you want to read it, okay, you can read it. And thank you for you. And I hope this revision is is benefit for you. And I hope I hope to you to take in or gain the high marks إن شاء الله. شكرا لكم. أشوفكم على خير. السلام عليكم ورحمة الله.